In this video, we'll talk about plasmid DNA isolation via the alkaline lysis method. Plasmid DNA is a really important tool in molecular biology lab. People who are cloning the gene, they need to propagate the gene in a bacteria by transformation reaction. If you want to know more about transformation, video is there in the I button or description. But anyway, propagating the plasmid, amplifying the plasmid, or storing the plasmid is really important. So overall, in this video, we'll try to understand how from bacterial cells one can chemically isolate the plasmid in a purified form. So there are two methods by which one can do that. There are column-based purification, which use the principle of affinity chromatography for purifying. And there is also chemical purification, which use fundamental chemical-based approach to purify the plasmid. Obviously, you can understand the first one is kit-based and customized, so this is more costly, whereas the second, chemical purification or alkaline lysis, is an inexpensive method. It's very cheap, but very efficient. So in this alkaline lysis method, you start with solution number 1. In a moment, I'll tell you the composition, but you first resuspend the bacterial cells by resuspension buffer. In the resuspension buffer, you have tris, EDTA. EDTA is very important because it chelates divalent metal ions such as calcium and magnesium, which are also important for DNases. So by chelating these divalent ions, you can also chelate DNase activity. Also, if you remove these calcium and magnesium, it would destabilize the membranes as well. Glucose is also added in this solution to maintain the osmolarity and ensures the cells doesn't prematurely burst. So overall, the resuspension buffer has a composition like this. So in order to prepare 100 ml of resuspension buffer or solution 1, you need to add all these components: glucose, tris-Cl, EDTA, water, and have to adjust the volume till 100 ml. Then the important part is the alkaline lysis. In order to take the plasmid out of the cell, you have to lyse the cell to take out the plasmid. And this lysis step is performed by solution number 2. This solution number 2 can be imagined as a lysis buffer, which contains sodium hydroxide and SDS. So obviously, by presence of sodium hydroxide, you can understand this particular solution is highly alkaline. And that gives us the name, the alkaline lysis. Anyway, the alkaline lysis step is performed by solution 2, whose composition is exactly mentioned here. But I'll tell you what exactly happens. After putting the alkaline lysis solution, the solution 2, you mix it gently and wait a little bit of time such that the lysis can happen. In this step, first of all, NaOH loosens up the cell wall, SDS pops hole in the membrane, and that releases the overall chromosome of bacteria and also the plasmid DNA in the solution. Now, also the DNA gets denatured because all the interaction between the DNAs, the base pairs, the hydrogen bonds are now disrupted because there is an alteration in pH in the solution, right? And this is not favorable for hydrogen bond formation because it is highly alkaline. So DNA gets denatured. Particular point of time, if you take the solution and open the tube, you would see strand-like appearances coming out of the tube. Now the next step is neutralization. This extremely alkaline environment has to be neutralized. And this neutralization is done by potassium acetate and acetic acid. Obviously, by putting acid, you can neutralize the base. So it forms a salt known as potassium dodecyl sulfate with the SDS, which is present in the alkaline lysis solution, and this is an insoluble salt. So after putting this solution 3, what happens is you see cardi precipitate like stuff appearing in the tube, which are highly insoluble. You can visually see them. So then you wait some time and then centrifuge them. Now, once you add the neutralization buffer, the waiting is important because you want to wait for precisely the time that would allow the circular DNA or the plasmid to renature, but it doesn't give enough time for the denatured cellular DNA or the chromosomal DNA to renature. So you have to ensure that, okay, renaturation happens only for the plasmid DNA. So the timing is critical in this step. After that, one should centrifuge them and precipitate these insoluble substances, the salt of dodecyl sulfate. So after that, what happens? The aqueous portion is taken out, and along with this aqueous portion, 0.6 volume of isopropanol is added to precipitate the plasmid DNA, which is supposed to be in this solution. Now, isopropanol makes the DNA unhappy. So isopropanol first forms bonds with the DNA. DNA doesn't interact with the water, so it's not anymore solubilized. It's now precipitated. After ultimately, it forms a pellet after isopropanol precipitation, and this pellet can be washed again by ethanol washing. So ethanol is added, the precipitation is again done. So ultimately, after that, the pellet is dried and resuspended in nucleus-free water, or let's say some tris buffer. This plasmid, once isolated, can be stored in -80 or -20 degrees centigrade for years. Now, how do you know the plasmid is good quality that you have isolated? So you can run those plasmid in a gel, and it should show you several bands. I mean, there are many outcomes possible, like bands correspond to nicked plasmid, linear plasmid, supercoiled, or circular, closed circular plasmid. The predominant form that you should be expecting is basically the supercoiled one, and that's why you should be getting the thickest band in the supercoiled region, and other bands should be very faint. Anyway, you can also check the quantity of plasmid, that how much is the yield of this isolation process. So one can put a 1 microliter plasmid there, and ultimately one can check the absorbance at 260 by 280 nanometer. It should come around like more than 500 nanograms per microliter for a good yield, and 8:260 280 ratio should be 1.8 for a pure DNA. If this ratio is not proper, then there could be a problem or contamination in your plasmid DNA. By looking at these parameters, you can understand how good the isolation is. You can find more flashcards and notes in my Facebook page, and you can find more notes in Instagram page. Anyway, you can support the channel via super thanks. You can use Paytm, PayPal, or UPI for paying. See you in next video.